Assalamu alaikum. Dear students, I am here Hafiz Muhammad Ammar, co-teacher for course Microbiology, code BOTN 4128. Today we are here for lecture number 21, entitled Growth of Microbial Cultures. In continuation of previous lectures, today we will discuss other methods including genetically transferred material and genetic recombination. When you hear the word clone, what do you think? Maybe of the dolly sheep or experiments carried out in microbiology labs. But it's true that the bacteria around you, around on your skin, in your gut, growing on your kitchen sink, they all are cloning themselves all the time. Bacteria reproduce by splitting in two via binary fission, as you already studied in the last lecture. Binary fission makes clones, or genetically identical copies of the parent bacterium. Since the child bacteria are genetically identical to the parent, binary fission doesn't provide any opportunity for genetic recombination or genetic diversity, except some random mutations. This is the contrasts with sexual reproduction. Still, genetic variation is the key to the survival of species, allowing groups to adopt the changes in the environment by natural selection. That's true for bacteria as well as plants and animals, so it's not too surprising that prokaryotes can share genes by three other mechanisms like conjugation, transformation, and transduction. The contents which will be covered today are the four different types of exchange of genetic information or genetic recombination. The genetic information carried in the DNA can be transferred from one cell to another. However, this is not a true exchange because only one partner receives the new information. In addition, the amount of DNA that is transferred is usually on a small piece of the chromosome. There are several mechanisms by which this genetic change takes place. Those are transformation, conjugation, transduction, and transposable elements. Bacterial transformation is a process of horizontal gene transfer by which some bacteria take up foreign genetic material like naked DNA from the environment. It was first reported in Streptococcus pneumonia by Griffith in 1928. In 1944, Avery demonstrated the transforming principle of the DNA. The process of gene transfer by transformation does not require a living donor cell, but only requires the presence of persistent DNA in the environment. The prerequisite for bacteria to undergo transformation is its ability to take up free extracellular genetic material. Such bacteria with this ability are termed as competent cells. The factors that regulate natural competence vary between previous genera. Once the transforming factor DNA enters the cytoplasm, it may be degraded by nucleases if it is different from the bacterial DNA. If the exogenous genetic material is similar to bacterial DNA, it may integrate into the chromosome. Sometimes the exogenous genetic material may coexist as a plasmid with chromosomal DNA. Reasons for transformation. The phenomena of natural transformation has enabled bacterial populations to overcome great fluctuations in population dynamics and to overcome the challenge of maintaining the population numbers during harsh and extreme environmental changes. During such conditions, some bacterial genera spontaneously release DNA from the cells into the environment, free to be taken up by the competent cells. The competent cells also respond to the changes in the environment and control the level of gene acquisition through natural transformation process. The phenomenon of transformation has been widely used in molecular biology. As they are easily grown in large numbers, transformed bacteria may be used as host cells for the following to make multiple copies of the DNA, in cloning procedures, to express large amounts of proteins and enzymes, or in the generation of complementary DNA libraries, or in DNA linkage studies. These are the applications of the transformation process. Here is the pictorial representation of transformation process. You can see in the picture right, there is red colored some foreign particle that is being uptaken by a competent bacteria. In the that foreign particle is in the form of DNA, stable persistent DNA, and this DNA taken by competent bacteria may integrate with its own genetic material. You can see in the right, or it may exist as a plasmid, coexist with its own genetic material. Here is another example. If plasmid being absorbed by a competent bacteria, that will coexist as a separate plasmid and bacterial own genetic material. Transduction. In transduction, viruses that infect bacteria move short pieces of chromosomal DNA from one bacterium to another by accident. Even bacteria can get a virus. The viruses that infect bacteria are called bacteriophages. Bacteriophages, like other viruses, are the pirates of the biological world. They command a cell's resources and use them to make more bacteriophages. However, this process can be little sloppy. Sometimes chunks of host cell DNA get enough inside the new bacteriophages as they are made. When one of these defective bacteriophages infects a cell, it transfers the DNA. Some bacteriophages chops the DNA of their host cell into pieces, make this transfer process more likely. The other group of prokaryotes, archaea, besides bacteria, are not infected by bacteriophages, but have their own viruses that move genetic material from one individual to another. Transduction is an efficient means of transferring DNA between bacteria because DNA enclosed in the bacteriophage is protected from physical decay and from attack by enzymes in the environment and is injected directly into the cells of bacteriophages. If you look into the picture at number 1, the bacteriophage is being injected in the bacterial cell where bacterial chromosome is already existed, and this phage is coexisting as with its own genetic material. In number 3, you can see different bacteriophages is multiplying in the bacterial cell. There are some virulent bacteriophages and some defective bacteriophages. and in this cell creates new phages including phage and host DNA material. While in 5 to 6 portion, the transduction phage inserts donor DNA, and donor DNA included in recipient's chromosomes due to recombination you can see in the number 7. In episomes and plasmids, transduction is not only confined to transfer of chromosomal DNA, but episomes and plasmids may also be transferred. Penicillin resistance in Staphylococci bacteria, the plasmids determining penicillin resistance in Staphylococci, are referred from cell to cell by transduction process. Genetic mapping of bacteria provides an excellent tool for genetic mapping of bacteria. Treatment of some inborn metabolic defects. It has also been proposed as a method of genetic engineering in the treatment of some inborn metabolic defects. Conjugative plasmids encode an extremely efficient mechanism that mediates their own transfer from a donor cell to a recipient cell. The process takes place in one direction since only the donor cells contain the conjugative plasmid. Conjugation is the transfer of DNA by direct cell-to-cell contact, that is mediated by plasmids, which is non-chromosomal DNA molecule. Or DNA is transferred from one bacterium to another after the donor cell puts itself close to the recipient using a structure called pilus. DNA is transferred between cells by this structure known as pilus. Conjugation is a process in which one cell, which may call donor or male cell, makes contact with another, the recipient or female cell, and DNA transferred directly from the donor into the recipient cell. You can see in the picture. This has been considered to be the bacterial equivalent of sexual mating in higher organisms, but in the analogy it is irrelevant, as following conjugation the female bacterium is in turn converted into a male cell. Conjugation, in contrast to transformation and transduction, requires contact between donor and recipient cells. In gram-negative bacteria, donor cells produce a specific plasmid-coded pilus, which is called the sex pilus, which attaches the donor cell to the recipient cell. Once connected, the two cells are brought into contact directly, and a conjugal bridge forms through which the DNA is transferred from the donor to the recipient, as you can see in the picture here. Many conjugative plasmids can be transferred between and reproduce in a large number of different gram-negative bacterial species. Plasmids may vary in size from few thousand to more than 100,000 base pairs, which may call megaplasmids. Pilus, that is only produced by donor cells, attaches to recipient cell and brings the two cells together. The mobile plasmid is nicked, and a single strand of DNA is then transferred to the recipient cell. Both cells synthesize a complementary strand to produce a double-stranded circular plasmid and also produce pili. Both cells are now viable donor for the effect. Certain structurally and genetically discrete segments of DNA which move around and between chromosomal and extrachromosomal DNA molecules within cells are called transposons. means jumping genes. This mode of genetic transfer is called transposition. These transposons vary from simple insertion sequences to complex. Transposons are larger, like 4 to 25 kbs. Composite elements contain genes for movement as well as genes that encode for various functions such as drug resistance, toxic productions. Unlike other processes that recognize DNA, transposition does not require extensive areas of homology between the transposons and its destination sites. Transposons provide a natural mechanism for the movement of genes from one chromosome to another. In 1950s, American geneticist Barbara McClintock discovered transposons in corn during her studies on maize genetics, and she was awarded the Nobel Prize for Medicine in 1983. In bacteria, transposable elements sometimes carry antibiotic resistance and pathogenicity genes, which make bacterial disease causing. If one of these transposable elements jumps from the chromosome into a plasmid, the genes it carries can be easily passed to other bacteria by transformation or conjugation. That means the gene can spread quickly through the population. The simplest transposable elements are insertion sequences, or you can say IS elements. It is a short sequence of DNA around 750 to 1600 base pairs in length. It contains a gene that codes for enzyme transposase, which catalyzes transposition. The transposase gene is bounded at each end by inverted repeats, sequences that function as recognition sites for transposons. The more complex is called composite transposon. All transposons contain the information for their own transposition. A transposon is a segment of DNA with one or more genes in the center and the two ends carrying inverted repeat sequences of nucleotide sequences. These are complementary to each other but in the reverse order. Because of this feature, each strand of the transposon can form a single-stranded loop carrying the gene and a double-stranded stem formed by hydrogen bonding between the terminal inverted repeat sequences. You can see in the picture right below here. Mechanism for amplifying genetic transfer. Transposition is a mechanism for amplifying genetic transfers in nature and has been identified in microorganisms, plants, and animals. Furthermore, because they may be carried between cells on plasmids or viruses, they can also spread from one organism or even species to another. Another significance is antibiotic resistance. Many transposons contain genes for antibiotic resistance. Transposons appear to accomplish in nature gene manipulations similar to the laboratory manipulations that have been called genetic engineering. Transposons are thus a potentially powerful mediator of evolution in organisms. In short, following key points we have discussed today. In transformation, a bacterium takes a piece of DNA floating in its environment. And in transduction, DNA is accidentally moved from one bacterium to another by a virus. In conjugation, DNA is transferred between bacteria through a tube between cells. Transposable elements are chunks of DNA that jump from one place to another. They can move bacterial genes that give bacterial antibiotic resistance or make them disease causing. These are the reference links or books from which the lecture was prepared. You can also study these reference books and links. Thank you very much.